So
Sweet.
hello everyone. Thank you so much for joining us for this unusually late lunchtime lecture. We're here today with Dr. Iroro Tanchi, who's a postdoc at the University of Washington working on ecological communities. As part of a research agenda to target historically understudied areas, she's currently focused on West Africa's Gulf of Guinea forests, where she studies insectivorous bat assembly structure through an ecomorphological approach to understanding resource partitioning and species integration. She also leads an NGO that works with local communities to protect critical forest and bat habitats in Nigeria while expanding research infrastructure in West Africa. She enjoys hiking and swimming. And I'm, yeah, thrilled to ask you to kick off this wonderful story about bats and where and how they forage.
Thanks, Khaled. Hi, everyone. I am really excited to be speaking to you today. I saw the numbers and I'm really shocked about how many people love bats. And I'm really curious to learn about bats. So I want to start off by thanking the Linnean Society for inviting me to give this talk. It's a really special honor for me to be able to share with you some of the work that I've been doing for the past almost decade plus, actually decade and a half. But anyway, so I I really want to kind of just start by saying that my research is really interested in understanding how there's so many bats and how they coexist, especially in a changing world. And that's really because tropical forest mammals are typically about 50% of them will be bats. So there's a high alpha diversity for bats across sites, and they occur almost everywhere except a few islands and Antarctica, and they're largely understudied. So that sort of just puts me in that space where I'm like, let's go do what nobody else is doing. But I'm a community ecologist, and so what I typically tend to do is to try to understand how species are distributed across space, especially space. Maybe not so much time, but I do tend to do a bit of time work as well. But when you're looking at how species are distributed across space, you tend to also look at the regional species pool. That is to say, what species are around those spaces that you're comparing or trying to understand. And then for me, I am particularly interested in the processes that give rise to what species are found where and what factors might be driving those. And I'm not going to talk about any of those processes today as such, but I'm going to just really talk about the actual work that I'm doing. And I want to kick it off by answering the first question that I get asked a lot. Why nighttime? Why do bats forage at night? And, when we first started sort of bat research, so I was thinking about this, the idea was, oh, maybe it's predator avoidance. So you avoid predators in the day, but some of the researchers suggested that's not the case. And oh, what about perhaps not having to compete, say, with birds? So the aerial niche, for example, is quite in the day, quite populated by birds, so to speak. But essentially, what the consensus now is that it is an open niche at night. So there's this huge buffet of food that bats have taken advantage of. So it's not so much one or the other on the left, but it's more the case that the niche was open and bats took advantage, and that's really exciting to know. All right, so just a little bit about how I got into bats. It started during a field course organized by the Tropical Biology Association. And I'm going to quickly say that this is a UK-based organization, and so I feel quite connected to the UK as such that there's a program that has come out of the UK that's supporting my research. So I basically was on this field course in Uganda and got told about bats, and I just got completely blown away. And I've been studying bats since. And I've gotten additional support from the Harrison Institute in Sevenoaks, Kent. And I also did a master's at the University of Leeds, where I was basically recording bat calls on both sides of the M5, south of Bristol, all the way to Essex. So, again, a lot of work with the UK over time and UK colleagues, and I feel really pleased to be able to be speaking to the Linnean Society members today. And that's kind of how I got into bats. So my MSc also was on bats, like I was saying. But essentially, what I have now come to do these days is to try to understand bat foraging more in the context of understanding the multiple dimensions of bat niches. And if you're thinking about bats, this is kind of what you think about. If you're thinking about how they forage, or at least the foraging niche, you're really thinking about where they're foraging. So that's kind of a habitat use scenario. And you're looking at, excuse me, how they do that. So feeding strategy, and then what they're feeding on, and that's the prey, right? But for most of the talk, I would really focus on just these two things because that's what's in the title. But my main research actually these days as a postdoc is now on that what part of it, which I don't really get to talk about that much. But when we talk about where bats are foraging, it's really the question about a spatial dimension, right? So if you look at this on this schematic here, what you're really seeing is multiple spaces in, say, around vegetation. You've got the open space, which is all the white bits, and then you've got edge space, which is all the sort of grayish bits, and the really dark gray is more cluttered space. And the whole idea is that bat echolocation is designed to be well adapted for each of these spaces. So they're well suited. So as you can see, some of these boxes here are actually echolocation calls of bats. So the signal design, how the echolocation signal is designed, that design varies from with each of these spaces. And essentially, I was trying to say this. I'm sure we all know here that bats echolocate at a high frequency beyond the range of human hearing. So humans will go up to about 20 kilohertz if you're a teenager. If you're not, you're probably doing something much lower. But bats would typically do, all up to say, I think the highest in a bat's been like 250 kilohertz. And that's basically a bat just the equivalent of a bat screaming out its lungs and just tearing up the space almost. But when we look at echolocation design, excuse me, we're really trying to say what the chord structure looks like in what's called a spectrogram. So this is on the left, on the bottom, you have time, excuse me, on the y-axis. So on the left here, you've got frequency. So these bats, this is a bat that occurs in the UK, Rhinolophus ferrumentinum. It collocates about somewhere around just about 70-ish kilohertz, and it basically does this thing where it's all pure tone except for these sweeps where it goes down and goes basically like a something like that. That's kind of what the bat's doing when it's making that sort of call. And those all of those things about, an upward sweep, a pure tone, and a downward sweep, they really carry information for the bat. A bat that's foraging at night, we use all that as well. And in fact, I like this spectrogram as well because it basically is showing that as the bat approaches the insect, so you get this call. These are all search phase calls. So the bat's like flying through and it just emits a call, and it emits another one. It's still searching, okay? But by the time that echo returns and it starts to pick up, it starts to race towards a particular target, in this case, the insect, it starts to do what we call a feeding buzz, okay? So it starts to echolocate really quickly. Remember the y-axis here is time. So the frequency at which it emits those calls starts to really increase because time really helps in localizing what object you're dealing with, in finding your prey, more or less. And so it increases that frequency of the emitted call. And so when you see this, when you're out recording bats, you basically know that it's about to snatch a bug, And it does that to the point where it's so rapid that it basically almost cancels out because it's now sort of ends because it's now grabbed the bug. But then when you also think about bats in this sort of foraging space, the other thing you're also thinking about is the wing morphology. And that's because the shape of the wing, how wide versus how long it is, what you call the aspect ratio, excuse me, really determines how much it's able to control its speed as well as maneuver in space. So if you're a clutter bat, you really need broad wings that allow you to really pack a lot of energy as you flap your wings, and so that you can kind of control slowing down because half the time you're really just in clutter. There are leaves all over the space, and you need to kind of avoid a lot of them. If you're an edge bat, you're kind of in between a clutter bat and an open space bat. So an edge bat will be something more like this, where it's not nearly as broad, but not nearly as thin as an open space bat. So you see how an open space bat will be much longer and thinner because they maximize speed. So they're really in the open space and just trying to forage for whatever is out there. So you really need to be fast. And long, thin wings are really useful for that. But, what it just means is if you're studying bats, ignoring these differences have really important implications. And so this is where I'm now going to take you away from the talk about the where bats forage to the implications of ignoring this and how that's gone into some of my research. Now, it basically starts with a map by this really nice gentleman, Finley, who was a curator at the Southwestern Museum in the US. That's in New Mexico. And he basically concluded that African bats are not very species rich, that essentially if you're here in southern Nigeria, you get 40 bats, but if you're in about the same equatorial range, so distance from the equator, if you're in say Malaysia or in Sumatra, you start to get different numbers. In fact, in Malaysia, you get about 100. In Sumatra, Sulawesi, sorry, in Sumatra here, you get about 80 bats. So that's about twice the number. And of course, the Malaysian number is even more than twice the number. And that result, excuse me, this basically gave rise to a dogma that, African bats are not diverse, and we know what that means. If an area is not very diverse, very few people want to go study things there, especially in the tropics. So that affects a lot of things beyond just the science. It also affects what the future science does. And a lot of people started to want to question this. In fact, started questioning this. A lot of people who've done work in Africa. And essentially, the story is that Finley at the time was really working with what he had. Basically, this was, in the 80s and 90s when people were sort of summarizing patterns across the world. And in fact, someone had done this also for birds around the world as well, especially tropical areas. And Finley was in Cameroon. He basically threw a mist net in the forest understory and only got 10 bats. That really makes you think, oh man, there are no bats here. But really, what the story is because of the way paleotropical bats are adapted to the forest understory. If you use a mist net, you're not going to get any bats in the understory. But what you really need to be doing in the understory in the paleotropics is to use a harp trap. Finley, by the way, has primarily most of his work was in the neotropics. So he was sort of coming from a neotropical background to this question about how many bats are in Africa. And in fact, at that time as well, at the time he published that book, actually, it's a book. Excuse me. At the time he published that book, harp traps had just been used, excuse me, in Southeast Asia, and so that didn't factor into that conclusion that Afrotropical bats were depopulated. In any case, we examined the literature in 2020, just around when COVID was taking off, and concluded that hop traps really drove a revolution in discovery of bats in the paleotropics. And this is actually just data from Southeast Asia alone, where just around when hop traps became really popular in the region, following work by Charles Francis and Tigger Kingston, who's actually my PhD advisor, that things began to take off in the species description. And by 2008, when some of the more recent work came off with some work from Pipat Soysuk, you basically start to see that harp traps contributed to more than twice the number of species descriptions that mist nets allowed. So just imagine if we were just using mist nets alone in these habitats, harp traps would really start to change things is what this turned out to be. All right, so just about when I was starting my PhD, paper was published by Hecht et al., and they basically showed that a lot of the Afrotropical diversity is clustered around mountainous areas. So here in the Rift Valley, up here in Ethiopia. And that really got me thinking, because I wanted to always just walk in southeastern Nigeria on the border with Cameroon, where there's a lot of mountains. And that's kind of where I then went off to ask this question, whether Finley truly was right. Because even though if you people had asked the question, they hadn't really addressed it by comparison. So when you want to address such a question, you do need to make such comparisons, right? So to begin with, my PhD was about elevational gradients and bats, and I wanted to, at least one of the questions was to address whether Afrotropical bats are truly depopulated. And so I conducted a lot of surveys, and a lot of it was against the background of work that Katrin McCain had done, where she showed that elevational gradients, species richness along elevational gradients takes three forms, and a lot of it was around some of the drivers of species richness. But essentially, this is the landscape I did my PhD in, and this star here, up here in Nigeria, you see all that relief that we're just kind of on the edge of that Cameroon volcanic climb, but we do get a few mountains that go up to about 1,800 meters. And so my work was in Afi Mountain Wildlife Sanctuary and Cross River National Park. And these are my sites. And essentially what I did was trap bats along 6 strata, each of those so I would have a site at about 150 meters elevation, and I would have 4 transects. Along each transect, I had 5 hop traps, and I could only do 10 hop traps a night, because at some point you sort of run out of the ability to check up to 40 traps at a site. So in any case, I always had 10 hop traps per night, and that would be between 2 transects. And so I would do that on each of those elevational bands, and I essentially also recorded ecomorphological traits, but also trapped insects and conducted vegetation surveys. And I'm not going to go into any of those, but I just want to show how just by using a lot of, doing a lot of hop trapping, I basically compared my data to sites in Southeast Asia that had similar capturing protocols where hop traps have been used quite a bit. And so I could compare forest understory bats with Southeast Asian, forest understory bats at my sites in Nigeria with Southeast Asian sites. And what I basically showed that besides the site in Malaysia where every 20 meters of a 3 kilometer grid square sampling area, I showed that my sites had similar, excuse me, uh, species richness as other sites in Southeast Asia. The Malaysian site is the most sample site to date in the Paleotropics, and you can imagine what that means. When you hypersample a site, you tend to get quite a bit of the diversity at that site. But anyway, I see that a lot of these also, some of these sites were also showing that they've reached the asymptote, so maybe nothing new might be added. But essentially, broadly speaking, Afrotropical sites are not that much different from Afrotropical sites are not that much different from Indo-Malayan site was what the study showed. And I wanted to ask a bit more about why. And so remember that when I started by saying I'm a community ecologist and you look at sites and you also want to look at regional patterns. And so I looked at regional level patterns. And essentially what I was seeing was when you when you take ecoregion data sets, ecoregion level data sets, and you examine how bat communities are similar or dissimilar between ecoregions, and you start to get nestedness data sets and turnover results, you really start to see that paleotropical, Afrotropical sites, so the lower Guinean forest is not much different from the Malaysian sites, but the main differences are really in how open space bats and edge bats are not so different between both sites. But then the real difference starts to come when you look at, say, edge species and narrow space bats. And it's really, if you actually look at this side of the plot, the biggest differences are really with nestedness, and because in the Malayan site you get, so Malay Peninsula, you get a lot of nestedness. So most of the sites are similar. So even though there are more bats in the Malayan site, the Lower Guinean forest did not do much differently in terms of total beta diversity and turnover. And so it's more regional processes that are driving that pattern that you see, excuse me, in the similarity between sites. And I showed this in this publication, but let me just point out some of the really cool things that I also recorded during my study. At least 10 new country records were recorded for Nigeria. 8 of those were caught exclusively in hop traps. I mean, imagine what a loss it would be to just be in this site and not be able to record bats that are truly in the forest understory. But also one of those species, forest understory bat caught exclusively in a hop trap, is a species of conservation concern and has now led to a huge conservation project. In fact, as far as we know, it's the first conservation project for an endangered species in Africa. And how that came about was a few days after discovering that species, trapping that species at the Afi Mountain Wildlife Sanctuary, our team ran away from a big fire that broke out. And this is the scene we saw when we looked back after getting down from the mountain in the middle of the night. And so this fire sparked a flame, quite literally, in us. And so we started this conservation project where basically farmers would set fires to their farm as part of not just slash and burn, but to maintain already existing farms. They would set fires to their farms, and basically those fires might escape if they're not well attended to. And in our case, what we then did was to work with the communities to set up an early warning an early warning wildfire prevention and response program where we first of all start by quantifying and predicting wildfire risk using weather stations like this, and we communicate it to communities using that. And then in cases where fires still break out after doing all of this, local community members will respond. And this is a big project. I mean, we have over 50 people employed on this, and they work every dry season to make sure that fires are not spreading from farms. And in fact, fires even on farms are also reducing, and that's a really good outcome for us. And what we see here is a map of our Field Mountain Wildlife Sanctuary, and the fire that pushed us out of the forest in 2016 burned through a good chunk of the forest, like almost 50% of the forest. But as soon as we started talking to community people and, helping with their their local laws and setting up, programs to support long-term wildfire prevention, we started seeing fewer fires and almost zero, and in fact, zero wildfires, except for the year COVID hit and when fires also went through that same area that we had fires in 2016. We basically have been counting the past 4 years now that we've got zero wildfires in the sanctuary, and next year, knock on wood, we're hoping will be the 50th year when we don't have wildfires. So we know that a lot of the problems we face in Nigeria are not restricted to Nigeria, and also actually similar problems are experienced in other parts of West Africa. And so we set up a West Africa Mammal Partnership to get colleagues together so that we can not only network and collaborate, but see how we can start to scale up some of these successful strategies we've used in Nigeria to protect bats. And we've got members from all over the place. Only a few countries are not represented at this point, and we're hoping to see more people in the future. But a huge part of that program is to support students and train students who study bats and other small mammals in the region. And this is our last cohort from last year. And for the first time, we had 100% retention of students in the program, because typically you get pulled in, you get to do a field course, and then if you're not if your project doesn't really meet the standard of the quality of what we're doing, you won't get further support. But this time, all of the students had good projects, and they have continued to receive support afterwards. And this is just a photo to show that, a lot of the work is both local and international experts training students in the field to really set them up to do their degree projects. These are postgraduate students, by the way. And part of that is also part of our work, really, is that we're working towards setting up a field station. And in fact, we've just started building one in the forest, and we're setting up another one on the Obudu Plateau, so that we can really start to document the biodiversity of this area, and also provide an opportunity to serve as a hub, more or less, to support students in the region. Now, that's all that about my PhD. That's all I would say about my PhD. That's not all the work I did for my PhD. But if you really come to the how question, it's a matter of foraging strategy. And with that, we're really looking at how bats pick up their prey when they're foraging. And so you have some groups doing aerial hawking, and this is the Rhinolophus ferromanginum, so the greater horseshoe bat in the UK, actually. And you get bats that glean prey from the surface. And so these bats going for this prey on a leaf or either on the ground, so vegetation or on the ground, or tree bark or whatever. But basically, it's prey getting grabbed from the surface, and you've got bats that also trap or pick up prey. So in this case, a fishing bat, for example, is trawling prey from the surface. And this, I can't remember, I think this is a nocturnal from the UK that's going for an insect on the surface of the water. So, there are different strategies that bats will employ, and a lot of these strategies, in fact, are tied to their echolocation. Excuse me. I'll drink some water. All right. So again, kind of going back to these multiple dimensions of bat foraging, what we're really dealing with is all of these sort of niche spaces, right? So there's a spatial niche, there's a behavioral niche, and there's a dietary niche, right? So when you start to think of it, what you start to realize is that um, you need to really combine each of these dimensions to get your bat in whatever space you're dealing with. And essentially, if you're at a local site, you have a bat in hand, say it's a rhinolophus, and what you're dealing with is a forest understory bat. So that spatial niche is what I described earlier about the where. So it will be a clutter bat. But when you're dealing with feeding strategies, it's more about how the bats get in the prey, right? And so you start to combine all these things because a greater horseshoe bat would be a forest understory bat, but the way it picks up its prey is by aerial hawking. Whereas some other rhinolophers might be picking up their prey by passive, so some sort of passive aerial hawking. So they basically hang out sort of like an ambush predator. They hang out and just wait for the prey to descend and, excuse me, and they go pick up the prey. So you've got to combine all these things to really understand what where your bat falls in within that space. But I guess why I pull this up is to really say that a lot of understanding around the dietary niche is still quite opaque because bats are really difficult to study, as you can imagine. You can't just except for bats that maybe roost in caves, and you have to find those caves first. You can't just walk into the forest and just grab bat poop, whereas you can do that for elephants, for example. All right, so that thought about the dietary niche being a tricky thing to nail down in terms of the categories and groups that you find in bats has really led people to ask this kind of question about, do we really have bat guilds? So in terms of diet, right? Because what we typically have is bats just get assigned to say, animalivore or frugivore, or if you're in the neotropics, you're really dealing with even a greater diversity because of all the ecological diversity. And bats in the neotropics, you start to have well, we'll have one carnivore in the paleotropics, but in the neotropics, you've got a few more carnivores. You get sanguivores and things like that. So are those really guilds though? That's the question. And some of the work that I've been thinking about in my postdoc is whether those guilds are even functional in the first place. And we know that there's been a lot of work with the functional axis of bat diet. And essentially, what we know is that there's a lot of, it has a lot to do with the cranial morphology. As you move away from longish, slim-faced bats that will typically say do nectar or have, say, a very soft bite, so to speak, you move towards bats that are short-faced, kind of like a bulldog, with really with a really hard bite and the skull's very robust. That functionality is not heavily well represented in bats, and as you can imagine, there are very few bats for which the functional diet have been quantified, certainly not in the Afrotropics, and that's where my work comes in. And so this is sort of a reverse way of showing you of what bats feed on. I should probably have put this at the very beginning, but I felt that because it tied in with this story, I brought it in here. So it's a true Bat 101. It's just Bat 101 towards the end of the talk. And so what do paleotropical bats feed on? We're looking at fruit and nectar. This is a cute nectar feeding bat that's in feeding on banana and nectar there. And I've caught that bat a few times, and it's really cool. Excuse me, the African morph actually. But then actually that's the bat I'm talking about, the megaloglosus. Excuse me. And well, others would feed on so in the paleotropics again will feed on primarily insects, but you get few doing fish, and there's one carnivore in Africa that will take birds and small mammals. But I put blood there just to kind of show, to kind of point out to people that yes, bats do take blood, but they're not paleotropical. So you're really looking at, what prey you get in those foraging guilds, so the spatial niche, and things will vary with what bats are doing versus what the insects are doing. And we know that insect size will scale with what bats are capable of eating. Excuse me. I'm not sure why this is just. All right. Well, so I'm currently also looking at whether that the relationship between bat prey and their bite force, so how hard they're able to crunch down to break up the prey, whether that's preserved across those foraging groups. And for my postdoc, I basically trapped bats across 9 sites in the Gulf of Guinea forest. So in southern Nigeria, I got 6 sites, and then along the Gulf of Guinea Islands, excuse me. I also trapped bats there, and I usually have a large field crew, thanks to a lot of people from SMACON these days helping out with that. And essentially, I would go out, trap bats, and have them bite on a couple of plates that are attached to a force transducer that's able to then be read off on a device that tells me how hard the bat's biting. I collect some of those bats as needed, and I take those into the lab and basically do a CT scan to extract the skull, and so not do it by hand. That takes time. I did that during my master's. But do the CT scan, collect the skull measurements, and then I go ahead to ask questions. But I just wanted to point out that this is usually a lot of effort. Collected trapped over 70 bats and collected lots of poop samples, and now doing a lot of skull measurements. And essentially, I'm asking whether what the bite force relationship is with prey hardness. And this is a big part of the research, trying to understand how prey properties match up to bite force in bats. And with that, I was just going to say thank you to all of the people who have supported me in the past. If you're interested in some of the work I'm doing, please watch my space. I can be reached here, and I've got a website that you can also follow updates on. And with that, I would like to say thank you, and if you have any questions, I'll be happy to take them. Excuse me.
First, I'd like to say thank you so much, Iroro. That was absolutely fascinating. And my camera doesn't seem to be working. There we go. And we do have a Q&A box full of questions. And I'll say to anyone else who has any questions, please pop them in there. We'll get to as many as we can. First of all, we have one. Someone's asking, what is a harp trap? Can you describe how they
Oh,
work?
this is great. I get that question a lot. So a harp trap is, it's sort of like, think of a harp, the musical instrument, and if you line them up, so this is how you sort of line them up that way. So you've got like four harps in a row, and then you string them up together in a frame. But this time is not a harp. Okay, so you get your string set of strings, and you line them up so that they're sort of slightly offset, the strings. And it's strung up in a 2 by 2 meter frame, below which, let me kind of run it that way while I'm describing, below which there is a bag, so that as um, the bat hits one set of strings, it will then slide down into that blue bag here. So yeah,
Ah,
and it can be quite heavy. And in fact, in my case, two harp traps are equivalent to one luggage allowance. So that's about 23 kgs, so about 50 pounds. So I logged
yikes.
about 10 of those up those mountains. It was always fun. I mean, I'm always grateful to people who either were helping or porters who were paid to carry those things. They're not light at all.
Gosh. Um, someone, same question. Do any of these traps risk injuring any of the bat species?
Mm. In fact, hop trap is much, well, how do I say much less? Um, the hop trap is less stressful for bats because what's basically happening is they slide down. So you're flying through that trail there, and you hit that string, you just slide into the bag. No injuries, nothing. And you just hang out because it's a cloth bag. So you hang out, bats basically the tiny feet just hold onto the bag like so. And then, you the scientist, you walk up to it and raise the flap and collect your bat. But mist nets can be quite difficult, stressful for bats because they tend to get tangled. And in fact, when you're doing especially mist net surveys, the best practice is to check your nets. It started by people used to say 30 every 30 minutes, but now it's like every 10 minutes, really.
Wow.
Because as soon as your bats are getting tangled in there, they're kind of trying to chew their way through. And they do cause a lot of damage to the nets because they got really sharp teeth, right? But then they also get so tangled sometimes. It's hard to get a bat out of the net. And so it's stressful for you, stressful for the bat. And actually, I think my first bat died in my hand
Mm.
as a beginner trying to extract it from a mist net. So I hate mist nets for bats. I just love my harp traps, but they're so heavy, and they're expensive too. You can buy 25 mist nets for one harp trap. That's kind of how they
Wow.
scale. But then they're also kind of nets are also kind of useless for understory bats too. So you really don't have a choice. You have to get your beloved harp trap. Yeah.
Um, on that note, we have a question asking how do they actually catch bats? Because if they have their sonar, they're wondering, yeah, can they avoid being caught by hearing the hop traps? Or they say, or
That's a
perhaps you
good
only catch certain varieties that are less able to detect nets, which is also interesting.
that in fact that's the term we use in our paper. We call bats that are able to detect nets mist net avoiders. So they
Ah,
avoid mist nets because they're able to see it real fast. Okay, so let's sort of back up a little bit. Um, Bats are not emitting calls every single microsecond, okay? So they emit a call, they're still in flight, right? And they wait for that echo to come back and they emit another one, things like that. So they keep doing that. And so some of the bats, when you're like, I don't know, maybe 2 meters or less than a meter from okay, in fact, let me sort of scale back a little bit again. So go back a little bit, right? Um, so the echolocation that forest understory bats use, they don't travel very far because they're usually high frequency. So high frequency calls attenuate very fast in the atmosphere and in clutter, they basically just disappear. So you have to be close to your object to see it. But if you're not emitting every single minute, or I shouldn't say minute, but you're not emitting every single moment, there comes a window where your trap is just in front here and you sort of ram into it. Okay? So that's one thing. Now, mist nets are because they're heavier material and you see how that sort of the pouch of the mist net sort of has increased the density of the net at this point. So they basically are very visible to forest understory bats that are clutter adapted. That is, they are very good at detecting clutter. So, but when you have a harp trap like this with all those strings, the strings are really tiny. It's like the tiniest fishing line possible. Okay, so they're really tiny. I can't remember the size now, but they're really, really tiny. Okay. Um, because they're set up in such a way that they're offset, so there are spaces between the strings, right? A bat that's flying thinks it's going to flip and fly between the strings, but then the next set of strings are offset. So the harps are not just lined up like that. They are lined up in such a way that they are offset to, so the gap it was going to fly through the first set of strings. That next set of strings offsets the first one, and so the bat hits that
Mhm.
next set of strings and slides down into the bag. So there are a few things going on there. It's about how much echolocation, how the distance of echolocation in the forest understory, of echolocating bats in the forest understory, um, the thickness of the material, so how detectable, how much how much echo it actually releases. But when the bat's even close enough to it, it's still organized in such a way that it tricks the bat. It's sort of what we call it funneling in the bat. The bat is funneled in, and so it doesn't have an excuse. It just hits the strings and boom, it's in the bag. But
It's
I
really
have to say
interesting.
that there's a study that looked at capture success, and harp traps are not necessarily 100, they don't have 100% capture success. And in fact, I can't remember where this study was done. It must have been the UK actually. Um, Bats were still able to avoid harp traps significantly. Okay, so bats really, you don't catch all the bats that come by your harp trap, is what I'm going to say.
Hmm,
Yeah.
pretty cool. Uh,
Hmm.
we have, oh wow, the questions keep rolling in. We're going to get through as many as we can, but we
Okay, I'm going to
unfortunately
be
have to keep
fast.
quite
All
to
right.
time today. So. Someone has asked, do you use acoustic lures to lure in bats to the traps?
We don't currently use those primarily because not a lot of them have been developed for African bats, but we've seen, successes in some areas of the world where people have used them, but we don't use lures in our work, not currently.
This person asks, how did you manage to continue working on bat conservation with local communities, and how did it go during COVID-19 with the bad PR that bats got at the time?
That's a good question. And I have to say that because of some of the work we've done in the area, we sort of were very, I guess, close to the communities already before COVID happened. And no, in fact, let me sort of go back a little bit. In 2014, when I started doing my field work in those communities, we just had an Ebola outbreak in West Africa. So when we showed up and said we're studying bats, people were just like, "Oh, really? Okay, I don't know the Ebola bats." That's sort of the language you hear. So we already started like chatting with them and letting them know, look, the risk is the risk is the chance of getting Ebola from bats very low, but is very consequential when it happens. But essentially what that meant is we're already doing a lot of outreach before COVID happened. So by the time COVID happened, it was much easier to keep saying, bats are not the bad guys here. We're the one destroying bat habitat and getting into more and more contact with bats. But let's not forget that these are communities that also hunt bats. And so it's kind of a two-way conversation where you try not to play up too much the risk, but you also point out the risk. And then they tell you, look, if you think I'll get, COVID from these bats or, Ebola, you're wrong because, my forefathers have been doing it before now. So it's somebody else who's gonna get the disease and not them. So there's that complexity in the interaction. But we're already doing a lot of outreach and a lot of engagement. And I think that helped a lot during COVID.
That's really interesting. Oh gosh, I'm trying to pick this one.
Sorry, am
No,
I
no, no.
rambling too
There's
much?
no. It's really. I think you're having exactly the right amount. People who are
Okay,
just really
that's good.
interested, so we have lots of questions.
Oh, that's
Um,
great.
this one I think is I'm curious about. If bats are, easily tricked by, well, maybe not easily, but tricked by nets like this, why don't they crash into other things, or do they?
Um, They do crash into other things. And in fact, we think, echolocation is this perfect system that's been set up. And let's start with when bats are exiting a cave. So when there's a huge colony of, millions of bats, for example, people used to think, well, that they don't bump into each other. But when sort of some of the camera trapping surveys and infrared camera surveys started coming up with a lot of their results, bats crash into each other all the time, like in such a but it really is that point about the fact that echolocation only goes so far. And when there's a lot of and you're not emitting calls all the time, you know. And it's a very remember flight is already energetically expensive. It's harder to fly than to walk, okay? Um, and then imagine talking non-stop because you're in flight. Okay. Imagine flying for a long distance and then talking nonstop. I mean, which I was speaking for 20 minutes and I would need to just have a drink of water just now, right? It's a really echolocation is not a simple process.
Hmm.
It's very energetically expensive as well, So um bats do crash into things quite a lot. And they do miss their way quite a lot. They're cute little things and they're smart and whatnot, but they make a lot of mistakes too because of, it's a really difficult process managing the whole shebang. You're flying and you're
Mm.
shouting at the top of your lungs and, it's difficult.
There's one here asking if, because they're saying in the UK there's bats that eat a lot of insects. Would you
Mhm.
expect the kind of bite strength and cranium shape to be different for a bat that eats mostly insects as opposed to a bat that eats nectar and fruit?
That's a good question. And this is where that point about food hardness is really difficult to just import it from the neotropical research programs that have focused on this. Because in their case, their diets across bats is kind of on a continuum, and there are a lot of bats that are omnivores. Okay, I
Mm.
mean they're echolocating bats. Some of them will have a combination of insects and fruit in their diet, whereas you move over to the new or old world, so the UK, Nigeria, Malaysia, all the stuff that you're dealing with are strict frugivores and strict nectarivores. The occasional fishing bat.
Mm.
Um, so that question is really tricky because it's not a concomitant um direction of change in fruit hardness versus insect hardness. So, fruit hardness for frugivores, say in Nigeria, would not be on the same scale as insect hardness for insect eating bats in Nigeria,
Mm.
or at least in the paleotropics. So the cranial shape is still going to do, because it's the laws of physics, right? It's still going to do what it does, which is, if you have a stoutish face, you're able to handle harder things. If you have a longish face, you're able to handle softer things, or not able, but that's what you're limited to. That's the
Mm.
point. Um, uh, but it doesn't scale from insect. So like across dietary domains, as it were. Um, but that question keeps me up at night. I have to tell you this because it's the reason I've had to invent a new way of measuring food hardness, and you'd find out eventually when I publish the paper. But I'm measuring food hardness in a different way from what they're doing in the neotropics, using the distribution of the food particles and the hardness of those food particles. And I'm actually even going beyond that and potentially trying to explore material properties rather than just the physical toughness of the material. But how all of those things would then scale with skull morphology between groups, that's a whole other thing. So I'm just sticking with my insects. In fact, for now, my forest understory bats. Keep it simple for now while we figure out what our bats are really doing. Sorry,
That's
am I
incredible.
taking up all the time? I
No,
have.
not at all. We have time for just one more question. I'm going to be cheeky and
Yay.
ask my own question, which is,
Okay.
do you have a favorite bat? And if so, which
Oh,
one
I
is
do.
it?
I do. I do. And if people think it's the bat that we're protecting, no, it's a cutie. I love it. It's a 7 gram bat that just turns into this beautiful thing when it's in flight, and it's got this really cute nose. But it's not my favorite bat. My favorite bat is the Lavia frons. This is the yellow-winged bat, and I had it. I actually made a costume for it this last Halloween. So
Amazing.
I had it. That was my Halloween costume. Anyway, so it's a yellow winged bat. It's the entire wing is yellow, and it's got yellow nose leaves, and the ears are yellow, and it's just really pretty to look at. Look, the reason it's my favorite is as a master's student when I was trapping bats. And I was trying to convince my rangers that bats are not ugly because they were like, why are you keeping us all night for these ugly things that
Oh,
smell so
no.
bad? And I was like, they're not ugly, they're not smelly. And then we caught one bat that smells it's got like a peanut butter smell. So I
Ooh.
was like, hey, peanut butter is not a bad, it smells like granite. And then we caught the last bat we caught that night was Lavia fronts, 2 individuals. Oh my gosh, the rangers could not believe their eyes. It basically they went from bats are ugly to bats are really cute. That's why it's my
Oh,
favorite bat.
changing
It
heart.
it makes it easy to sell bats. I guess that's the point.
Oh, well, thank you so much for that. That was absolutely incredible. And I
Thank
encourage
you.
everyone to go off and Google that bat and have a look at it. I know that's what I'm going to do. Um,
Thank you so much, everyone. This was so much fun. Thanks.
as usual, check out our upcoming events. They are on our website, and we'll be sending an email follow up later. And keep an eye out for when this shows up on YouTube shortly. Thank you so much again, Aurora. That was absolutely amazing.
Thank
I look
you.
forward to seeing you all again soon.
All right.
Bye.
Bye.